Hi, my name is Jonathan King, and this video is created for the Principles of Synthetic Biology class at UC Berkeley. My project is about the pulse-mediated self-deactivation of a toggle switch. In this video, I'll describe the development of a genetic toggle switch model that deactivates after a predetermined number of toggles. My design is advantageous in that the time it takes to deactivate can be tuned by altering the degradation and binding affinity of a single repressor. Before I go into detail about my system, though, I'd like to speak about the motivation for my project. One of the most important things to consider when designing synthetic biology solutions is safety. In order to assuage the public's concerns about releasing transgenic organisms into the environment, scientists often implement fail-safe measures into their designs that limit unforeseen and potentially harmful effects. If a genetic circuit was set to expire after a certain amount of iterations, then one could be confident that any adverse behavior exhibited by the system wouldn't last forever. One medically relevant application of such a circuit would be in gene therapy. Imagine that a patient's cells have been engineered to deliver a treatment via genetic toggle switch. In this instance, my self-regulating design could prevent an overdose of treatment by limiting the number of times it could be delivered or toggled in a set period of time. Finally, as the title suggests, my design incorporates the pulses of an incoherent feed-forward motif to limit the number of times the toggle switch may operate before it eventually fails. The following is the genetic regulatory network that describes my system. I begin with the inducible toggle switch developed by Collins et al. That is able to switch between the states of high LAC-I expression and high TET-R expression by adding the inducing molecules IPTG or ATC. I then tied LAC-I to the start of an incoherent feed-forward motif. In this motif, LAC-I both activates and represses AZ. If both LAC-I and AY are necessary for AZ's expression, then AZ will only be expressed in a short pulse when the system moves to the high LAC-I state. In order to deactivate the toggle switch, I then used AZ to repress TET-R. This means that after so many iterations of the toggle switch, switching from LAC-I to TET-R, AZ will be at a high enough concentration to fully repress TET-R and force the system to remain in the LAC-I state indefinitely. This design is based on the assumption that AZ can be reliably tuned to slow its degradation over time, so that a threshold concentration can be used accurately. In addition, its binding coefficient can be modified such that a desirable level of repression of the system can be achieved. In the genetic circuit implementation of my system, I've incorporated both real and hypothetical genetic parts. For instance, I've introduced two hypothetical repressors, R1 and R2, as well as the activators A1 and A2. The promoter, called PA12, acts as a hybrid promoter that only activates transcription when both A1 and A2 are present. Because I was interested in the delays of transcription and translation for my incoherent feed-forward motif, I chose to model the mRNA dynamics of my system. My design incorporates several transcription factors, 3 of which are repressors and 2 of which are activators. Each binds with its own binding constant to the transcript with a matching binding site. Note that Kr2, the dissociation constant for R2, is what creates the threshold for the inactivation of the toggle switch. I've hypothesized that modifying this parameter will directly affect the number of iterations that the toggle will function correctly. The next set of chemical equations on the right describe how each transcript is translated. The first transcripts are constitutively translated when no repressors are present, while the last, MAZ, can only be translated in the presence of both of its activators, A1 and A2. I also modeled the degradation of mRNA and proteins in my system. For simplicity, I've assumed that each mRNA will degrade at the same linear rate. In addition, I've chosen the degradation rate for all proteins to be the same, with the exception of R2. Since R2's concentration directly affects the deactivation of the circuit, I chose to model its degradation separately. I can experiment with different hypothetical modifications to slow down its rate of degradation and maintain my circuit's ability to fail after a number of iterations. To be more precise, if I wanted the toggle to fail after n iterations when R2 reaches the threshold concentration of X, then the degradation rate must be slow enough so that R2 does not drop in concentration by more than X over n throughout the entire experiment. If this holds true, then the number of times that the switch has been toggled can be inferred from the concentration of R2 at any time. In addition, I've modeled how the inducer's IPTG and ATC limit protein function by writing the following sequestration equations that describe how each inducer is assumed to bind irreversibly to its target protein. In the following chemical equations that describe protein production over time, I have made the assumption that each transcript is transcribed and degraded at similar linear rates. In addition, the concentration of LAC-I and TET-R depend on their sequestration by IPTG and ATC respectively. I'll now describe the mathematical models for my system. The following equations describe the nitty-gritty detail of my circuit and how each transcript is regulated by transcription factors. These two equations help modulate the toggle switch portion of my circuit. The first describes how MA is negatively regulated by TET-R and degraded linearly. The second describes how MB is negatively regulated by both R2 and LAC-I and is also degraded linearly. Each term here in parentheses is called a Hill term and determines what fraction of maximal transcription, or KTXN, is possible given the concentration of transcription factors in the cell. These next two equations describe the incoherent feed-forward motif part of my circuit. The first describes how MAY is negatively regulated by the repressor R1 and is degraded linearly. The second describes how the expression of MAZ is reliant on both the presence of A1 and A2, while also describing its degradation. In order to analyze my system, I used the biosystem library within MATLAB. I added each species as a component of the biosystem before describing their relationships using my mathematical models. At first, I wanted to make sure that I could model my incoherent feed-forward motif and generate pulses of R2 that would build up over time. As you can see in the diagram on the right, successive pulses of A1 expression resulted in pulses of R2 expression. Remember, I've assumed that R2 has been modified to avoid degradation within the time frame of the experiment. After confirming that my model of the incoherent feed-forward motif was accurate, I combined this model with a toggle switch as described by Collins et al. Then, I simulated several pulses of my inducer molecules IPTG and ATC, causing the system to toggle between states. I expected to see my toggle switch lose its ability to express TET-R, thus disabling its bimodal stability. However, in my first few attempts, I was not able to express TET-R at all. After re-evaluating my circuit and its parameters, I realized that the repressor I was using was bound too strongly for TET-R to ever be expressed. The solution, I realized, was to lower the strength of R2's binding to the TET-R transcript. This graph demonstrates the circuit's behavior after decreasing R2's binding strength. It's now clear that each time the toggle switches states, TET-R's transcript is more poorly expressed, thus limiting its ability to repress the LAC-I transcript. This in effect destroys the toggle's original behavior. LAC-I only continues to dip in the graph because of the presence of IPTG. Without IPTG, the transcript containing LAC-I will be able to be expressed, meaning any other proteins on this transcript will remain in high concentrations indefinitely, or at least until R2 degrades enough to re-enable the expression of the TET-R transcript. In conclusion, with adequate adjustment of the binding affinity and degradation rates of AZ, also known as R2 in my implementation, my genetic circuit is able to perform its self-regulatory behavior as described and should prove useful in synthetic biology contexts. The safety mechanism it provides might interest other synthetic biologists who would like their circuit to reliably fail after several iterations. I hope that my work demonstrates the utility of incoherent feed-forward motifs in order to regulate other genetic devices like oscillators or combinatorial logic circuits. In the future, I'd like to experiment with other behaviors of AZ. Perhaps instead of using it to disable the toggle switch by targeting TET-R, it could target other members of the system, or simply act as a molecular signal that the circuit is at the end of its life. Lastly, AZ's repression of the TET-ARC transcript could also be improved by using a repression cascade, increasing the digital-like behavior of my circuit. Thank you.